Hi, I'm Professor Davis, and I'm going to talk to you about the scientific process. The natural world around us is full of amazing things, and science is one way that we can study it better to understand the world and our relationship to it. While we should not exploit nature for, when I use our, you, I'm talking about the collective, while we shouldn't exploit nature for our collective, our desires, it does present opportunities for problem solving for some societal need. We're going to see this theme that pops up over and over that often nature has the solutions. Science is, in general, a systematic process for understanding the world around us. It's a set of steps and procedures to follow to keep it consistent. Science also refers to the accumulated body of knowledge, and that's often what is referenced when we hear it in mainstream. Well, science says this, scientists say that. It's a collective, accumulated body of knowledge. Again, it's also a good tool to address societal needs. Science can be descriptive, so processes that aren't fully understood yet. Observations, recording, something to better understand a system that we don't know fully yet. On the left is a picture of Jane Goodall, and probably familiar with her work with chimpanzees. And that's also a picture of one of her journals. She is a major figure in the world of animal behavior, introducing the world of chimpanzees to the public. And she had to go through a lot to get to Africa to do her research. And she's certainly a model, not just for women, but anyone in the field of science. She spent decades observing and describing their behavior and their social structure. So this is an example of descriptive science. But it can also be hypothesis driven. Maybe there's a specific research question in mind, maybe a specific problem to address. For example, after the Deepwater Horizon oil rig explosion, oil flowed into the Gulf for about 3 months, and that had long-term effects on the environment. In order to find out the extent of the damage, scientists are looking at specific parts of the environment. For example, how deep into the sand did the oil seep? So this is a specific research question around which an experimental design is created. So we have the scientific method, which you probably have heard of, maybe played around with it in previous classes. So the idea is if everyone uses the same process, when scientists come together, they can talk about their findings in a way that can be compared. Some fake studies have been found out to be fraudulent because of this. A study came out of China about reverting adult stem cells back to being embryonic stem cells. But when other labs tried to recreate the findings, it was found out that the methods and result of the original study had been doctored. So this is just a funny representation of the steps which we will go through each one. So the first part is about making observations. What about the natural world do you find curious? Is there some natural process or structure that you wish to explore more? And this was from my master's work researching how wolves and bison interact during a hunt. And I actually learned more about bison defensive behavior than I thought. So, for example, why is the bison running towards the wolf? Why is the wolf running away from the bison? This is different from what we would expect. So this may actually be the hardest step in formulating the research question. So we now form this based on the observations. We want to settle on a specific topic and develop that question to try to answer. A lot of times we're going to have many ideas and it's hard to narrow it down to one question. So hang on to those questions. They can be revisited at another time, but we can only test one question at a time. Often we'll do what's called a literature review, that perhaps somebody has already worked on this or worked on something similar, maybe in a different setting. So we may not have to start from scratch. We may be able to build upon what someone else has already done. From there, we formulate the hypothesis. So you probably have heard of this, an educated guess, it's often called. Well, it's a tentative answer to the research question and needs to be a rational accounting for a set of observations. So why do you think it's going to be the way it is? So let's say a medical study is testing a medication that will lower blood pressure. Simply saying, yeah, sure, it'll work, isn't enough. Well, why will it work? What is it doing in the body to make it work? We're going to look at a plant example. We want to test a fertilizer on plant growth. Our working hypothesis says that this fertilizer will increase plant growth compared to plants that do not get this fertilizer. Well, sometimes we use what's called a null hypothesis. You'll see a subscript 0 after the n. And this is opposite of the working hypothesis. So the null hypothesis says there will be no difference in growth in the plants getting the fertilizer compared to plants that do not get the fertilizer. Basically, that fertilizer has no effect on plant growth. So why bother with the null hypothesis? Well, it's easier to run traditional statistical tests from that perspective. So then we're going to design an experiment based on the predictions of the hypothesis. This is to help answer the research question. And we're going to manipulate and measure certain variables. So some of the variables we change, like the fertilizer or the soil, and that's what we mean by manipulate. Some variables we measure, and in this case, how well the plant grows. We have other types of variables, independent and dependent variables. In this example, independent variables are the kind of soil, fertilizer, compost, water, and light. And the way I remember it is to help give them personality. So light, soil, and water are going to keep on being light, soil, and water, regardless of what we do to them. They're independent of anything we do. The dependent variable is the growth of the vegetables or the plant that we're using. How well did they grow depends on the conditions of light, soil, and water. Now we only want to measure one variable at a time. So let's keep going with this example. Let's say that besides the fertilizer, we also vary the amount of water they get and the amount of sunlight that they're exposed to. When we look at plant growth, how would we know if it was the fertilizer, the water, or the sunlight that contributed to the plant's success? So if we want to test how much water contributes to better plant growth, then we need to leave the soil conditions and the amount of light the same. Likewise, if we want to test the amount of sunlight, we leave the water and soil conditions the same. We also need to be clear about what we mean by plant growth. To say something like these conditions help the plant grow better is too vague. Will we measure plant height? How about its mass, the number of leaves, the number of tomatoes, beans, or turnips that they yield? We need to be very clear about what we are doing. We have different groups as well as variables, and you might have heard of the control group. The control group provides us with a baseline to compare the experimental results to. Did the variable we are testing actually give us results? And in this example, if the group is getting hair loss treatment shampoo showed no difference compared to the group getting the regular shampoo, then we know the treatment doesn't work. Most medical research is done this way to prevent bias. The mind is powerful. For example, the placebo effect. People will respond to treatment even when they aren't getting it. They have done studies about the placebo effect where the subjects know they are getting a placebo, maybe a sugar pill or saline injection, and they still feel better. The mind-body connection is amazing. You may have also have heard of blind studies. And this is a style of research to help prevent bias. So single blind means the study subjects don't know what group they're in. So they don't know if they're getting the actual medication or a sugar pill. A double-blind study means neither the subjects nor the people administering the treatment know what group they're in. So if you don't know whether you're getting the sugar pill or the actual medication, neither does the person who's handing you those tablets. You don't want to inadvertently be influenced by the attitude of the person administering the treatment. There's somebody who has the official key with the subjects and what treatment group they're in. And so later they'll come back to that and see what the effects of if there were any effects of the medication. During this time, we collect data, and we'll look at a couple different types of data, mainly two types. I'm going to go back to our plant example. Quantitative, so think quantity, those are numbers, counts, times. So with our plants, maybe we're measuring the height, maybe the mass, the number of leaves, the number of vegetables yielded. There's another type of results called qualitative, think quality. Not that one's better than the other, but these would be observations, surveys, interviews. And in our plant example, maybe what shade of green the leaves are, what shade of color the vegetables are in. Another example using plants and qualitative results, you're probably familiar with Tabasco sauce. It is made in Louisiana from peppers grown there. The farmers use a wooden rod painted a specific shade of red to compare the peppers to, and that's how they know when they are just right to pick. And that's also where we get the name of the city, Baton Rouge, which means red stick. There's another little chart here for you to help compare qualitative to quantitative, whether something is expressed as a number, can it be counted, and examples of different types. So let's say we run our plant experiment with 50 plants in the experimental group and 50 plants in the control group. Having a chart of all that data is cumbersome and doesn't tell us much. This is what we call raw data. Nobody wants to look at that. And it doesn't show us any kind of relationship among the variables. Did that type of fertilizer actually help or not? So we're not going to use this when presenting our results. What we're going to do is take our data and kind of what I call digest it a bit. We're going to organize it into graphs and diagrams, maybe tables, and this will show relationships among the data, tell us the story of what the data is saying. So in this example here, where this is a bar graph, and bar graphs represent different categories. So in this case, we have plants that just got soil, plants that got fertilizer, plants that got compost, and then some plants got both fertilizer and compost. And that is along the x-axis, the horizontal line there. We would have the independent variable, always goes on the x-axis, and we include labels so we know what each bar represents and units. But in this case, it's just a description, so we don't really have units for that. On the y-axis, that's the one that runs along the side, and this is where the dependent variable goes. Also, they need labels and units, and in this case, the label would be plant height, and we do use the metric system in science, so that would be centimeters. And so from here, we can see the relationship among the soil type and how much the plant grew in height. This is a much better way to see the relationship between plant height and what type of soil treatment that they got. So now we're going to analyze the results, and yes, that often means statistics, right? Everyone gets scared by that. Honestly, statistics are an amazing tool to help us see, did we actually see a pattern? Do we actually see a difference? Or did we get the results due to random chance alone? And in the most statistical results, we look at the p-value. It's a probability value that, yeah, we did see the pattern, or no, we just got this due to chance. So let's say the average shoe size of a biology class this summer is an 8. And last summer's class average shoe size is a 9.5. So can we say that the students from last summer have larger feet? Or is a size and a half really all that much different in size? Well, we can run a statistical test that will tell us if there really is a difference. In science, we often use terms like positive relationship, negative relationship, and I just want you to be aware that positive does not always mean good. Negative doesn't necessarily mean bad. What this is referring to is a trend line. So in this case, we're comparing variables to each other. And on the left, that's a positive relationship because as one variable increases, so does the other. And we see that upward sloping line. On the right-hand side, that is what we call a negative relationship, meaning as one variable increases, the other actually decreases. And we see that by a downward sloping line. So, for example, the colder the temperature, the more clothing you may wear. That's just the term negative is just how we would describe that. Now, we need to be careful about correlation and causation. Just because it shows something is related does not mean one actually causes the other. There may be something going on. So, for example, the blue line here represents ice cream sales, and we can clearly see they increase from the months of May to it's probably about August, and then we see them decrease into the winter months. If we look at the number of shark attacks, that follows the line very closely. Now, is that saying that sharks are more attracted to people that have eaten ice cream? Or that when we eat ice cream, we make foolish mistakes about where to swim in the water? No, one does not cause the other. It just happens that ice cream sales go up when it's hot and more people go swimming when it's hot. So while we see a correlation, does not mean one causes the other. So that's something we need to be careful about when we're hearing about a group or a person presenting new findings and how things are related to each other. Now we're going to relate our work back to our hypothesis. We can only say that data supports or fails to support it. And it's okay if it doesn't. That's how we learn and grow. It's more interesting to find out things that aren't the way we expect them to. So it's okay to go back and say, well, maybe we need to revise our hypothesis. There's nothing wrong with that at all. In this section, we're also going to discuss the results. What do they actually mean? How does the type of soil affect plant growth? How does it answer the research question? We also need to take into account anything that may have influenced the results. So let's say we grew our plants in 3 greenhouses. And one weekend, a fan broke in one of them, and the temperature was much higher all weekend because there's no air circulation. This may have affected the growth of the plants. There's no intentional bias, but accidents, stuff happens like that and will still affect the results. So we call those confounding factors. We never say prove. We never say our data proves the hypothesis. Our results can only support or fail to support the hypothesis. We have to leave the door open for advances. Our technology, which we use as our measuring instruments, our understanding, those will improve. So we're going to get a better picture of how something works. Now you need to be careful because prove is often used easily in the press. Doctors prove that red wine and dark chocolate are good for your heart. That's not the whole picture, but it makes a good headline. So let's talk about an easy example here. For example, your flashlight doesn't work. So the question might be, why doesn't your flashlight work? Well, my first thought goes to the batteries are dead, but that's not the only thing that makes a flashlight functional. So the light bulb could also be burnt out. Both of these hypotheses are testable. So let's follow through testing hypothesis 1, that the batteries are dead. So we predict that replacing the batteries will fix the problem. So our actual experimental design is to replace the batteries. But when we check our results, the flashlight does not work. It does not support that hypothesis. So let's go with the other route, that the bulb is burnt out. So fixing the light bulb or replacing the light bulb will fix the problem. So we actually go ahead and do that. The flashlight now works. The hypothesis is supported. Now, we can't say that anytime any flashlight doesn't work, we've proven that it's the light bulb. We also have to be careful about what science can and cannot be addressed by science. A hypothesis must be testable and falsifiable. For example, saying that ghost fooled with the flashlight cannot be tested. This is not a judgment on whether or not you believe in the supernatural or paranormal. It just doesn't have a place within the scientific method realm. So let's look at a real life example. When we think of a giraffe, we probably think of a generic coat pattern, probably the Nubian or reticulated pattern. but there's actually 9 coat patterns, all within the same species. Then we also have the Labrador Retriever, but yet we are familiar with black labs, yellow labs, or chocolate labs. So the point here being that color patterns vary in nature even within the same species. So this one took a look at 2 populations of white-footed mice. Don't worry about pronouncing that. But they have different color patterns and they're found in different environments. So our study site was off the coast of Florida on the Gulf side. And you can see on the left there, that's what the beach looks like. Very light sand, sparse vegetation, and the little mouse there has a lighter coat. The inland mouse on the right-hand side, you can see the vegetation in the soil are redder brown, and you can see the little mouse there also matches that color pattern. So we need to think a little bit more about our hypothesis. Certainly camouflage comes to mind. What would support that? Well, foxes and owls are the predominant predators in these habitats, and they are very visual. And so it stands to reason that they depend on their sight to be able to pick out their prey. So camouflage would make sense. If the mouse blends in with its background, it is harder to be visually spotted by the predators. So keeping that in mind, they developed a formal hypothesis that said mice that did not match their habitat would be preyed on more heavily than mice that did match their surroundings. So what they did was they built mice models, painted them to match the surroundings, and placed equal numbers of each type of model in each habitat. It is highly unethical and unfair to take the little white mice and just kind of chuck them in the brown vegetation area, and to take the little brown mice and chuck them on the sand dunes. That's unethical. So what they're doing is they're making little representations, models of the mice, and putting them, both kinds, in both habitats. Then they recorded the signs of predation. Now I would ask, why would they bother putting inland mice models in the inland habitat and beach mice models in the beach habitat? Well, these are acting as control groups. Maybe the predators avoided the models for some reason. Fox have a great sense of smell, and perhaps the glue and paint that they used on the models would deter them. Now, owls and birds in general don't have a great sense of smell, except for vultures. You can think on why that would be. So anyway, the control group helps to account for any other reasons the models may not be preyed upon. And let's take a look at the results. So it turns out the potential smell of the models, that was not a problem. Let's look at the beach habitat first on the left. The light models, those are the ones that are camouflaged, and they are the control group. And they were preyed upon a little more than 25% of the time. The dark models are the experimental group, and they were preyed upon about 75% of the time. Does this support the hypothesis? Yes, it does. Now let's look at the right at the inland habitat. This time the dark models were camouflaged, and they are the control group, and they were preyed upon a little less than 25% of the time. The light models were the experimental group and were preyed upon a smidge more than 75% of the time. Does this support the hypothesis? Yes, it does. So can we conclude that mice who are camouflaged against their background are preyed upon less than those that stand out against their background? Yes, we can. Did the scientists prove anything? No, they did not. Okay, so then what happens? Well, it would be great to publish the work. That way you can share your ideas with the scientific and academic communities. And journals are peer reviewed, meaning that other professionals in that field read the study and they are a part of the publishing process. So let's say we want to publish our plant study. We might submit the paper to the journal Botany, which is the study of plants. Other scientists who study plants would read our work and decide if it's good enough to publish. It is a vigorous process, a lot of editing to do. The paper might outright be rejected, or maybe you need to do some edits before they'll accept it. It's like running a gauntlet there. So that's why there's that little cartoon there that it feels kind of brutal. I just want to give you some examples of some journals related to environmental studies that one might want to get published in. One's about innovation and societal transition. Just put Journal of Environmental Science, Informatics, Education, Sustainability, Communication, and Development. So hopefully this gave you a bit of an outline and a base understanding of the scientific method and the process that we use to inquire about the world around us.